graduate from Electric Electronics Engineering Department from Middle East Technical University, and now
Biraz mikrofonla ağzınıza yaklaştırır mısınız?
And now I am a master's student in Istanbul Technical University, and I am very excited to be here today to share our study with Okatan, my professor. And our title is a comparison of truncation thresholds and osmosis thresholds in predicting behavior from spike band extracellular neural recordings. Our presentation has 5 key sections. First, we'll begin with the introduction to stage R study, and then we discuss data that used, method that employed, and then we will dive into the comparison between the truncation and OTSU. And then later, lastly, we'll wrap up with the conclusion. And now let's start with the introduction part. Spike band extracellular neural recordings provide us a wealth of information from multiple neurons. And amplitude thresholds are widely used to detect the spikes from those complex data. And if in our pursuit of extracting the maximum information, the optimal amplitude thresholds methods becomes crucial for us. And in this presentation, I will highlight 2 significant amplitude threshold methods, and now let's look at them closely. Truncation thresholds inspired from the statistics, and it comes from truncated probability. OTSU-based thresholds comes from image processing, and actually it comes from the OTSU thresholding method. In the simulated data, truncation thresholds shows that estimate the standard deviation of subthreshold data accurately. And later research shows that OTSU-based outperform the truncation thresholds to estimate the standard deviation of subthreshold data. Recent research shows that mean and standard deviation of subthreshold data of the truncation thresholds are good predictors in the real data, and also based side actually there is a gap exists, and our motivation is to find that. Our question is, are mean and standard deviation of subthreshold data of also based? Are they good predictor in real data? If so, are they better than the truncation thresholds? And now let's discuss the data. Data is gathered from the rat, which is in the plexiglass cage wall, and there is in front of the cage wall there is a robotic arm with rotation axis and guided LED on top of it, and there are 2 levers near the top, and this top allows a rat to interact with the setup. And there are 2 light setups, 2 of them is in the cage and 2 of them in the direction of the rotation. If the rotation axis goes through the right direction, then the corresponding right LEDs will illuminate. Conversely, left lights will light up. And now let's look at how the data is gathered. The rat has freedom to walk in the cage wall whenever it wants, wherever it wants. And whenever it's approached the top and walk through the top, then there is an infrared beam to initiate the start point of the data. And then rotation axis choose one direction, in this case right direction, and the right LEDs will light up. The red analyze and choose one of the levers and press. Whenever it's pressed, then it is going to be a response point for us in the data. And if it's correct answer and correctly press the lever, then it is rewarded with water. This whole process is called trial. And to analyze this whole process, 0.5 time series are extracted, and this is called epoch. And these start points and response points help us to label the epochs. For example, these 4 epochs are pre-start epochs, and this epoch after the 0.5 time series later is post-start epoch. And to summarize the data, for one trial we have 8 epochs, and it has the rat is processed this trial 8 different times, either blue or red, and this gives a data structure for us. In the x direction corresponds to the epoch, and the y direction corresponds to the number of trials, and colors represent the right or left. And there are 16 different electrodes, and we also use 2 different features, mu and sigma. Although there are 16 electrodes and 2 different features, the whole process is same and the data structure is same. So, as a representative sample, I will continue with the 1 electrode. And let's continue with the method part. The method part first, we analyze how mu and sigma feature extractions. In the truncation thresholds, while calculating the truncation thresholds value, we have to do some truncated normal distribution, and mu and sigma values are obtained as the maximum likelihood of the truncated normal distribution. On the Otsuba-based threshold sites, this is a direct and Swift method, so we can more go to this part. Consider an epoch, and each epoch has time series in it, 0.5 time series in it. And we also took this epoch, divide the data into positive and negative parts, and apply the also-based methods, and find thresholds for each part. And then these two parts are merged. And after this merge, we extract mu and standard deviation from those data. And also this is time-consuming process, so we also store time value to find the truncation, to find the thresholds for each epoch, for both Otsu and for both Otsu and truncation part. So the data structure was like matrix time and matrix form, and the first 4 column corresponds to the pre-start point, and later corresponds to the post-start point. And our question was try to predict the behavior from those data. However, before the start point, there shouldn't be any information in data because there is maybe left or right trial info. So the question is there a significant difference in mu or sigma between pre and post-start categories when animal press either the left or right values? Is it statistically significant? And the data split into the pre-start and post-start categories and left and right. And we give this data to the Kruskal-Wallis test and to K-Kramer multiple comparison test to test whether statistically significant difference. And the answer was yes. So it means that pre-start categories mu or sigma do not change significantly with left and right trials, so we can throw them. But post-start categories has information of the left and right trials, so we can continue with them. So modeling the behaviors. This aligned to our motivation. Can we predict the behaviors, in this case, pressing left or right, from mu or standard deviation? Is it statistically significant? Can we predict the behaviors? So we train logistic regression with the 80 trials. And it gives probability continuous value output to us, and it corresponds to likelihood of pressing the right, because right was 1 and left one was 0. And these continuous variables transform into the binary value with thresholds, and this and then in order to calculate accuracy, we check with the actual true values, and we calculate accuracy for one electrode only. However, we have 16 different electrodes for both methods and for two features, and it gives 16 accuracies, and these are corresponds to the accuracy vector for us. And now we can compare two methods. When we look at the median accuracy values, if we use mu as a parameter, as a feature, truncation threshold has 90, Otsu base threshold around 82. If we use sigma value, then 95 versus 91 is represented. So it seems like the truncation thresholds outperform the Otsu based. But can we conclude this conclusion? Can we jump to this conclusion? So we test our question now. Does the accuracy change significantly with the thresholding method? In this case, truncation versus Otsu, and mu versus standard deviation. So is it statistically significant? This is the question. So we took this, we take this accuracy vectors and apply Kruskal-Wallis test and Tukey-Kramer multiple comparison test, and the answer was no. So we cannot state them one is better than the others. Sorry. So we also have time values for each epoch. We also compare this time, although it was significant that Otsu is calculated faster than the truncation. We also test this, and the answer was yes. Otsu based could be computed much faster than the truncation. And let's continue with our conclusion. What was the motivation? Our mean and standard deviation of subthreshold data of Otsu based good predictors in real data, and the answer is yes. If so, are they better than the truncation? No, difference is not statistically significant. And in addition to those two motivations, we also claim, maybe also check that Otsu-based thresholds are faster than the truncation thresholds. So, thank you.
Thank you.